Hi everybody. Today we are going to be talking about a great example of structure meets function, as well as a great example of quaternary structure. So today we're talking about myoglobin and hemoglobin, both transporters of oxygen in the body. This is found in Chapter 7, Section 1, so this is just the first half of the chapter. Although, yes, we're only covering half the chapter today, you will find that we're going a little bit more in depth than we have in previous chapters. So, I'm sorry this figure, for some reason, isn't coming out very clear in the video. However, hopefully on the PowerPoint slides that you have, you'll be able to see it a little better. And, apparently I left out my learning objectives, so I will get those written out for you as well. You'll see that in the PowerPoint, but probably not in the video. Okay? So, myoglobin and hemoglobin. So, as I said, both transport oxygen in the body. And they do this utilizing a heme prosthetic group. So, a prosthetic group is a non-protein group that is utilized in the protein for function. And we'll talk more about the heme in just a minute. Myoglobin facilitates oxygen diffusion from the blood to the muscle tissue. As we know, oxygen is poorly soluble in water and aqueous environments, so binding to myoglobin increases this solubility and facilitates diffusion. As you can see here, myoglobin is a monomer. It contains 153 amino acids that comprise 8 alpha helices. And these helices, although it's hard to see here, are labeled A, B, C, D, E, F, G, H. Okay? Now, hemoglobin transports oxygen throughout the body in the blood. So we find hemoglobin in erythrocytes, or red blood cells. Remember, we find myoglobin in muscle cells. Hemoglobin is actually a tetramer comprised of 4 myoglobin-like subunits. As you can see, each of the subunits in hemoglobin look remarkably similar to myoglobin, although they are different primary sequences. And hemoglobin is actually a dimer of alpha-beta protomers. So you have 2 alphas and 2 betas. So you have alpha 1, beta 1, alpha 2, beta 2 to make up the hemoglobin. And we'll talk a little more about that later. So let's get a little more in depth in myoglobin structure. As we can see, myoglobin, we have 153 amino acids that we already said, 8 alpha helices, and the heme prosthetic group that coordinates an iron 2 atom in the center. So here's our heme, and there's our iron. You can see it's coordinated in the middle of the heme ring. We have our N-terminus and our C-terminus. And then we got, our alpha helix, the alpha-beta beta helix. Then we have C, D, E, F, G, H, so on and so forth. So let's talk about the heme. So the heme is actually also called a porphyrin that has an iron coordinated by the 4 nitrogens of the pyrrole rings. So here's one of our pyrrole rings. You see we have 4 nitrogens at the corner of each of the iron 2 atoms. Now, iron 2 likes to actually have 6 coordinations, so the other 2 coordination sites on iron are occupied by histine on helix F. Okay, so it's the 8th residue in helix F, which happens to be a histine that we can see here. And the other site is coordinated by the oxygen molecule that it carries. The oxygen and the porphyrin are further stabilized by valine 11 on helix E and phenylalanine on helix, the CD helix bend. Okay, so these are hydrophobic groups that actually help stabilize the heme structure and also sort of trap the oxygen. Now, when the myoglobin is breathing and oxygen is coming off and in to the heme, these will actually swing out to facilitate the oxygen moving in and out of the molecule. The histine, as you know, actually has a hydrogen bonding, or not hydrogen bonding, but it's got a nitrogen with a free electron pair that helps coordinate with that iron atom. Okay? Now, when oxygen is not present, we have a water molecule instead. Okay? That coordinates in that center. Alrighty? So, interestingly, free heme has a 25,000 times greater affinity for carbon monoxide than for oxygen. Now, when the heme is in myoglobin, that affinity is reduced to only 250 times greater because of histine E7. The steric effects induced by the histine forces linear molecules like carbon monoxide and O2 to bind at the heme at a 120-degree angle. Now, although oxygen doesn't really mind this bend, carbon monoxide does not tolerate that bend very well. The carbon monoxide would prefer to come in perpendicular as opposed to 120 degrees, and that's why we get this reduction in affinity. However, as we can see, carbon monoxide still has a 250 times greater affinity for binding than oxygen, and this is how we can get, why did I put CO2? It's carbon monoxide, right? So this is how we can get carbon monoxide poisoning, because oxygen cannot come in and displace the carbon monoxide if the carbon monoxide concentration gets too high. Now, what's interesting is binding of oxygen alters the myoglobin structure. When oxygen is not present, the iron atom is drawn out of the plane due to its interaction with the histine F8 by 0.055 nanometers. Now, when oxygen comes in and binds, it actually will pull iron back into the plane of the heme, which also pulls on the histine, which then pulls on the helix. Now, we'll talk about how this effect, this structural effect, has a profound binding effect of oxygen to hemoglobin later on. Now, the binding of oxygen to hemoglobin is important. As we can see, it's a hyperbolic curve. Okay? This occurs when ligands interact independently with their binding sites. So let's say if myoglobin had more than one binding site for oxygen, let's say it's binding site 1, 2, 3, 4, 5, whatever, binding at this site does not affect the binding here, here, or here, or here of any additional oxygens. They will all bind equally from the first. Okay? What's also interesting is the P50, or the pressure at which 50% of the myoglobin is bound to O2, is 2.8 torr. Right here. So the P50 in arterial blood is 100 torr, while for venous blood it is at 30 torr. So at either arterial blood or venous blood, oxygen has a very high affinity for binding to myoglobin. So, and we'll see how this plays a role in the transfer of oxygen from myoglobin, or from hemoglobin to myoglobin, transferring oxygen from the blood to the muscle where it's desperately needed during very active metabolic processes. Okay? But as you can see, anywhere in the body, this is 30 torr, right? So this is the venous blood pressure. We will see that we have at least 90% binding of oxygen to myoglobin, or 90% saturation. So now let's talk about the structure of hemoglobin and how it compares. So hemoglobin is a tetramer of 2 alpha and 2 beta subunits. One of each of the subunits forms an alpha-beta dimer, and then the 2 dimers form our tetramer. Okay, so we have alpha, beta, and alpha, beta. Okay, I'm so sorry that this is hard to see in the video. Again, take a moment and look at your PowerPoint. But as you can see, we have myoglobin and we have our alpha subunit and our beta subunit, and they're all very, very similar. The alpha-beta dimers interfaces involve 35 residue interactions, a lot of them hydrophobic, but we also have some ionic interactions and some salt bridges occurring. And then cross interactions between alpha-1, beta-2, and alpha-2, beta-1 has 19 residues. Now, interactions between alpha-1, alpha-2, or beta-1, beta-2 are very minimal at best. Now, when oxygen binds to hemoglobin, remember we have that structural shift. At first, if there's no oxygen, the iron is pulled out of the plane of the heme ring. But when oxygen comes in to bind, iron is pulled back into the plane of the heme, which pulls on the histidine, which then pulls the helix. Now, in myoglobin, we really didn't see too much of an effect of that, but what that does in hemoglobin is that causes the entire alpha-beta dimer to rotate 15 degrees with respect to the other dimer. And this global structure has a crucial effect on the binding of additional oxygens, as we will see later. So here, remember, we have our deoxyhemoglobin. When oxygen comes in and binds, we get this global shifting of one of the dimers to create our oxyhemoglobin. And we'll see later that this is also called a T state and an R state. And we'll talk about that later. So what we find when we look at the binding curves of oxygen to hemoglobin, we see that oxygen shows a cooperative binding to hemoglobin. So remember, here is our myoglobin binder, right? It's hyperbolic. Well, we see instead a sigmoidal curve when oxygen binds to hemoglobin. And what this is indicative of is cooperative binding, which means when we have one oxygen molecule binds, it increases the affinity of another oxygen molecule to bind. So that's why first we see a slow slope of one binding, but once we start to bind more oxygen, we see that slope increase and we become more like myoglobin. The P50 for deoxyhemoglobin is 26 torr, while the P50 for oxy3, so you got 3 oxygens on the hemoglobin, is 0.2 torr. So that we can see that the 4th oxygen binds with 100-fold greater affinity than the first oxygen. And we were able to determine this using a hill plot. And you can look over the math of that. But basically what a hill plot does is it manipulates the data so that if there's no cooperativity, we see a straight line, as we do in myoglobin. If there's positive cooperativity, as we see in hemoglobin, we see something like this. If there was negative cooperativity, we'd see the opposite, right? We see the slope decrease. So as we can see, if we look at our P50 of this first asymptote, okay, again, we see that we have a P50 around 20 torr. And then if we look at our second asymptote, after we've bound 3 oxygens, we see that our P50 is dramatically decreased for binding that 4th. So that's how we were able to generate that data. So how does a shift in the structure change the binding affinity? So remember, as I said, the hemes, well, I guess I haven't said this yet, but the hemes in hemoglobin are 25 to 37 angstroms apart. So how can the binding of one oxygen in one of the hemes affect another part of the molecule over 25 to 37 angstroms away? And the reason is because the binding of one oxygen molecule to one of the hemes causes that overall structural shift from the T, which is that deoxy, to the R, which is the oxyhemoglobin. Now, this first initial shift is actually unfavorable because we break interactions between, now there's limited interactions, but we do break some of the interactions between our B parts. Our beta subunits. And once we've already broken those interactions, we've actually caused this huge global shift from this 15-degree shift that breaks those initial interactions between the two beta subunits. Then we have primed the other hemes for binding because we've already overcame that unfavorable shift in the global change. So any binding of further oxygens is no longer impeded due to the breaking of these favorable interactions between the beta subunits. Now remember, there were few of them. It was very few, and you can see in the chapter it goes in a lot more detail about the specific interactions and the specific ion bridges that are occurring that get broken that y'all will read about. Let's see. What else we got? Oh, okay, so this is really cool. So hemoglobin and the Bohr effect. So as hemoglobin transitions from the T state to the R state, the pKa of some of the amino acid residues is decreased, again because we're disrupting the ion pairing between some of the stabilizing subunit interactions. So we disrupt those ion pairs, which isolates those amino acids out of their initial microenvironment, and it makes their pKas more acidic. So what this means is at physiological pH, hemoglobin actually releases about 0.6 protons per oxygen molecules bound. Increasing the pH or decreasing the proton concentration will then drive hemoglobin to bind more oxygen. Because if you look at our equation, if we say hemoglobin plus oxygen yields hydrogen, if we decrease the if we increase the pH by decreasing the proton concentration, we will then drive the equilibrium further for hemoglobin to bind more oxygen. So this is just an equilibrium problem. So this will allow, increasing the pH will allow then hemoglobin to bind more oxygen at even low oxygen pressure. So this is important in capillaries, where the partial pressure of oxygen is low. Protons can be generated by bicarbonate formation. So remember, we're talking about the bicarb buffer in the blood. So if we form bicarbonate, we can produce protons. All right? So the protons generated by bicarbonate formation can then be taken up this way by hemoglobin, which stabilizes the T state over the R state, which then allows release of oxygen. Uptake of protons also increases bicarbonate formation, right? Because if hemoglobin is taking up the protons, then we're going to be forcing bicarbonate formation. The T-state hemoglobin can also bind and transport carbon dioxide to the lungs. So let's see if we can look at this. There we go. Let's see if we can look at this step by step. Okay? So let's start in the lungs. We inhale. We bring in oxygen. We have a high partial pressure of oxygen in the lungs, so that high partial pressure facilitates the binding of oxygen to hemoglobin. So all the hemoglobin is going to get bound by oxygen. We then enter into the bloodstream, and we're kind of going through the body. Now we're at the muscle. So the hemoglobin is going to donate its oxygen to myoglobin because we're at very low partial pressure. So the affinity for oxygen is higher for myoglobin than it is for hemoglobin at low partial pressure. So now that hemoglobin has gotten rid of its oxygen, now we're just floating around back in the blood. We're going to make our way back to our lungs. So we got free hemoglobin entering in the blood. It's taking on protons. All right, and these are protons formed by the blood buffer, the bicarbonate blood buffer. This uptake of protons, as we can see here, is actually going to cause more bicarbonate formation, facilitating the blood buffer. Hemoglobin can then also donate its protons, as we can see here, or hemoglobin can bind carbon dioxide, not that it's shown here, which can then bring it to the lungs and release it. Or, in the same token, hemoglobin can either uptake protons. This makes it look like it's donating a proton, but by doing that, by donating its protons, it's actually forcing carbonic anhydrase to transport, and this gets a little more complicated. I think I need to go back to the textbook on this. I am even getting confused by this, you guys. But I think the important thing to know is that the Bohr effect helps facilitate oxygen donation to myoglobin as well as transport of carbon dioxide to the lungs. Okay? So the CO2 produced by respiratory tissues diffuses from the tissue to the capillaries. This dissolved carbon dioxide forms bicarbonate only very slowly by the reaction. However, in erythrocytes, there's the enzyme carbonic anhydrase that greatly accelerates this reaction. In capillaries, where the pressure of oxygen is low, the proton generation by bicarbonate formation is taken up by hemoglobin, stabilizing the T state, which then induces hemoglobin to unload its bound oxygen. The proton uptake, moreover, facilitates carbon dioxide transport by stimulating bicarbonate formation. Conversely, in the lungs, where the partial pressure of oxygen is high, oxygen will bind hemoglobin, disrupting the T state pairs to form the R state, thereby releasing the Bohr protons, which can then recombine with bicarbonate to drive off CO2. So that's what this is. So as hemoglobin comes back to the lungs, oxygen will then bind, force the hydrogens off, which then allows the bicarbonate with carbonic anhydrase to release water, because we have those protons released from hemoglobin, so we get the release of water and CO2 in the lungs. So that's that end part. Now, another cool thing is a molecule called BPG. BPG also stabilizes the T state, so that's the deoxy state of hemoglobin. Now, BPG binds and stabilizes the T state by cross-linking the two beta subunits. So we have cross-linking at 143 and 143 of both of the beta subunits. What's interesting is fetal hemoglobin is a dimer of 2-alpha gamma instead of beta. Now, the difference in gamma is that it has a serine 143 instead of a histine 143. So doing this mutation here and here is by disrupting these histines in the beta subunits to serines, we disrupt the BPG binding. And what this does is this makes fetal hemoglobin have a higher affinity for oxygen than maternal hemoglobin. So now oxygen is transferred from the mother to the fetus by the facilitation of this mutation to make gamma instead of beta subunits. That's pretty cool. Now, another mutation that's not as cool is in sickle cell anemia. So this is actually a single amino acid mutation where glutamate 6 is mutated to valine 6 in the beta subunit. So we have a very hydrophilic, charged amino acid getting mutated to a very hydrophobic amino acid. What this does is this causes aggregation of the deoxy state of hemoglobin. And when you get this, because you have this hydrophobic interaction, you get this hydrophobic cross-linking between multiple hemoglobins, and it aggregates into a linear strand, which causes that sickle shape in the cell. So here we can see the valine, we get this hydrophobic pocket interaction between the valine 6 mutant and phenylalanine 85 with leucine 88. So you get a very hydrophobic, sticky, kind of call it like a sticky end. So this is what makes the cells have this very sickle shape. It causes a lot of clots. It's actually pretty painful, apparently. And even people who are heterozygotes, so they carry the recessive and the dominant trait, so they're not true sickle cell. However, their hemoglobin is about 40% hemoglobin S. So they have about 40% of the sickle trait hemoglobin, which they have a normal life. However, their erythrocytes have a shorter lifespan. So their blood cells, their red blood cells, do not last as long, which can cause severe anemia in these patients. Okay, so that's all I had for you guys today. And on class, the next time we meet, we're going to work in our workbook on problem solving with hemoglobin and myoglobin. So hopefully that will reinforce some of the material I discussed. Make sure you read the chapter. It's a lot of really good information. There is a good bit of math when we talk about the binding curves and the math that goes into that. I don't expect you to memorize the math, but what I would like you to do is to be able to recognize the plots, the hill plot, and then the binding curve, and then to tell me the differences between myoglobin and hemoglobin in both spots and how they indicate cooperative binding. Okay, so we will answer any questions that you may have in class, and I will see you then. Okay? And am I talking to myself the whole time? Let's see. Hopefully not.